Okay, sorry for the late. So today we have a lot to go through. So first, we will try different random number generators using the function, and then try to simulate single-cell RNA-seq data expression, so matrix. Then from there, we will try to see how can we create a function. So this function will put your command line together, so next time you don't have to type them in again, and you can change the parameters. Then we go through some descriptive statistics, compute the mean, variance, and coefficient variance in the single cell matrix. Then we will look at this covariance function, and then follow the slides and to compute using examples for variance covariance matrix. If we have time to revisit this dot product genomic interpretation, we will do it. And at the end, I will show you how to do this differential expression analysis in single cell data using the interface. All right, so let's start with this, and make sure you're in the working folder. In my working folder is documents, then MATLAB. So there's nothing in this space. There's no variable in the workspace. numbers. So in this case, if you do this histogram again, so you start to see the program smart enough because you have enough samples for each bin. It starts to create denser bins and allow you to see the distribution of this samples frequency within this each bins. So in theory, this should be the same frequency. You got the idea? Okay? For any given variable, so if you have a list of numbers, you want to see the distribution, then you do histogram using this function to visualize the distribution. Now let's do another. So if you do random n, so the only difference between this round simulation and compared to this is because there's n here, indicate we are using a function will generate normal distributed random numbers instead of uniformly distributed. And then if you do histogram again, you should see a normal distribution histogram. So if you do this again, you see those numbers are centered around the mean, which is a 0 here, okay? And with a standard deviation equal to 1. Again, for this, because it's centralized, it generates a centralized, have a mean equal to 0 all the time, so we don't need to give this random number generator function any parameters. It's always set the mean equals to 1, okay? So if you want, say I want a mean to be 35, then you have just 35 plus this number you get. You shift the mean to the 35, everything changed. Any questions for those? That's fine, right? So now we are going to look at the negative binomial distribution. By the way, before we move to there, I would ask, can we use any of the random generator we just described, the uniform random number generator and also normal distribution to simulate any of this data? Can we? For example, if you try, for example, you believe this is a follow the normal distribution, maybe you want to try create this and plus maybe 18 something to create. So I can give it a try and see whether we can create something similar to this particular highly expressed gene. So what I can do is to generate this number, but also plus 18. Okay, so this make this average move to 18, and this have some variation, and because the variance is there. So if I look at my number, and then there's 18, and then if I take floor, so this will make the value from this double precision digital decimal number into a integer. So it looks like I have something like a 17, 18, 19. But this start to, it's an integer, it's become a count, but it doesn't look like something like this with a 34, 24. The variation is much larger than this real data, right? So that's the problem. So if I did that, this variation is much bigger. So I only shift the mean from my simulator is not enough. I need to find a way to expand the variation in order to make this range much larger. But I know even I change the variation, I still not be able to capture those highly variable change of that. So this is a inherited problem with this normal distribution. It's not really good estimation of the real single cell count data. So people don't use, we just don't use this normal distribution to simulate those. So we, as I said, we use negative binomial. So we use this function instead of RAND-N. So we use X, we are using negative binomial R and D. Okay. Now I'm generating just one gene's expression, so 5,000 cells. But remember this, this function requires input parameters. So you cannot say, okay, I just wrote 5,001 there. Give me 5,000 simulation of this gene expression of that. That's not enough. We need the parameters. The parameter means we need, for instance, if I use this particular example, we need two parameters. We need 7 and 0.3. Or we need 1, 0.2, 10, 0.6. So we need, I just use 7, 0.3. So this particular gene, I'm going to simulate this gene's expression in 5,000 cells. And this gene's expression is given by these two parameters. And then we can see what the number looks like. So if I do this, now my number in X, I don't know if you can see, the first one is 24, 29, and then 6, 9. Looks like it's better than the normal distribution. Normal distribution, if I have a very narrow variation, like at 17, 18, 19, that's all around the centralize this average. But now this one seems I have a 35, 9, 15, so it's slightly variable than normal, and then because the parameter will decide the average and the distribution. So these two parameters you can change. So for another gene, this could be 1, 0.2, depends on the probability of that gene you want to simulate. So if you change these two numbers, parameters, you change that gene's expression profile. So let's see if we can try do another gene, so Y. So this gene, let's try the parameter here, 1, 0.2, okay? So what's the Y looks like? The Y, it's really small. I don't know how to make this font bigger. But the second gene, I also simulate 5,000 cells for that particular gene's expression. Then you see 8, 1, 0, and you start to see 0 because this Y is small. So this looks more realistic if you compare the real data. Let's just move forward to another. So I simulated another gene, have a 10 and 0.6 as a parameter. So let's see, gene Z have a 10, 0.6. All right, this is hard-coded. So I just imagine I have 3 genes. They all have a different parameter combinations in negative binomial distribution. So I simulate 3 and the same number of cells. So I can put them together and make a big matrix. See, I have a gene, single-cell gene expression data matrix with 3 genes in 5,000 cells expression. So how can you make the big X? So that's, you put them together. X, Y, Z. Okay, we create a single cell data simulator. In this 4 lines of code, I simulate 3 genes expression in 5,000 cells. And my output is a big X. If you look at the big X, it looks like a real data. Okay? If I don't tell you, you may think, okay, this is I extract from a real single-cell RNA-seq experiment results. So that's the purpose of simulation. You do this. So you may want to create more. So if you want to create 2,000 genes expression, you may have X1, X2, X3, X4, so you can do a loop. Right? You know how to do the loop to concatenate them together to make a big one. It just repeats itself. Well, when you repeat itself, you want to have a way to do these combinations of these 2 parameters. That's another question. So how can you change this every time you add 1 gene? Okay, so what's your idea? So your idea could be, I can generate random number using, for example, uniformly distributed. I can generate number from 0 to 1, and then times 10, then take integer, so I can make a random number here. I can generate a random frequency or p-values here, also uniformly. So in that way, you build another random number generator to give the parameter this, so it's 2 layers. You generate a random number and put it in here. You generate a random number, put it here, and then use this 2 random combinations, generate a random expression, and then try again. So this is 2 layers. Make sense? Okay. So one function put output gave to another function as a parameter. So in that way, you simulate the whole parameter space. Okay, so I'm going to show you how to use histogram to plot this distribution, put 3 together. Okay, you know how to do 1, so because you can do histogram, and then so this is the first one, our X, And then you want to hold. So if you want to, say, plot another one, but add another one onto it, so you do a hold on. So basically, this tell you, please do not flash away the previous plot, but I'm going to superimpose my second layer of these figures into the first one. So if you do that, you can continue with Y, and you start to see. So the blue one is the first, which is our distribution of X, the first gene, and then the second gene is yellow. And the first gene have an average is higher, and then the second gene is lower. So if you look at the average, this is close to 1, 2. Second gene, the first gene expression is higher, right? So you can do this for the third gene by doing Z here, and it keep adding onto it. If you can see, there are 3 PDFs, 3 curves. Blue is from a gene X, orange from a gene Y, and then this yellow from a gene Z. Okay? So if you compare this with the one in our slide, they're the same histogram. Okay, histogram, and to show you the distribution you want to observe from your variable, your data. Then different parameter generate different type of genes expression. So now we are going to create so this is our simulator. We are going to create a function so we can copy this code and put in the function. And so next time we don't have to type that again. We just run directly from the file. So this file is called any name that makes sense to you. So it's my script, for example. So if you do edit my script, this edit will create empty text file. So this my script M file will be saved in your folder, your working folder. So make sure you know where you save. My working folder is under the documents, MATLAB. So that's all my function, all my script are there. So if you do this, you you see there's a new file created with extension and with .m. .m is script MATLAB code file. And then you can copy that 4 line of the code and paste into here. So next time you don't have to type again. So I will also put all the histogram in there. So I copy all this, Control C, and put in this file. Here I need a function called a figure to pop up a new new plot. Otherwise, your first one, second one, all will be put together. Okay, but this is create a new one, and then you start to add things on. Okay, so once I finished, I can save. Now I have my first script file. So this is MATLAB code saved in my working folder now. I can clear all, clean this. I can close my script. So everything is closed, and just like you restart your machine, restart MATLAB, you will get something like this. What we haven't lost is the script file we saved in the folder, working folder. So we still have that in there. So the next time you can just double click and to open that script, and then click this run here, and you will regenerate what you had before. Anything you tried in your command window, which you believe is valuable, you should copy and save somewhere. So next time you can rerun it, just like retype them in. But for the long script, long code, you don't want to retype them. Here we regenerate XYZ and put them together. We regenerate this figure to show the histogram, the distribution of those three genes expression. Any questions? No problem? You can make it? Okay. Now we will move one step further. So this script is just one time. It's static. There's no parameter changes because we cannot change those parameters. You have to open it, change it manually, and save and run. But we want to make it into a function, so every time we can call that function and generate those differently. Let's see, the first function I want to make become variable is 5,000. We will still generate 3 genes expression, but each time sometimes we want to generate 1,000, sometimes 50, sometimes 50,000. So I want to make this become a variable. So I will make a function and make the number of the cells to be changeable. So I need to build a function. So edit my function instead of my script. But it looks very similar to each other. You still copy what you have here into your function. Well, I'm not going to plot anything. I don't need the figure for this function. I just need to generate the data. So now your script has no difference from your function. So you can still run this again, and you will have X, Y, Z, and big X in your workspace. All the same. The only difference is I delete those figures, so you don't see any figure change. Okay. 2 script files. Now I'm going to make this into a function. This function gave a name. You have to have keyword to indicate the script below. It's not a static, it's a function. So I'm going to make this function have output. For example, my output is X equal, and then I have function name. So I'm done. So MATLAB require your function name is the same as the M file. So you have to be named the file the same as the function. So because this is called my function, so I have to have this. And I can put so here means what this function require. What's the input? What's the output? Output will be the big X. The input will be nothing. So I don't need anything. I want to generate a matrix expression X. So now you can save this. So this is your first MATLAB function. Why you want to use a function? Because you want to, you are lazy. You want to keep the most valuable algorithm, which is this. But you want to be make a parameter allow you to change it later. But for now, we just have a very simple function, and you can call it and to generate X matrix. So now with this function, I can call, okay, this function give me X, so my function. Because you have your function defined, and then my function become available for you to use. And this function is very simple. It requires nothing, but it generates a matrix with 5,000 cells, 3 genes. Okay? And you will see there's a internal intermediate XYZ small variable will not be given in the workspace. It's internal, it's encapsulated into the function itself. So that's the inside or inner variable. It's closed in your function, so you don't see them, but you see the output. The output is your interface from the X, big X. Okay. You can run this function again. Say I can create another X2. So you call that again. This generates another one. And you don't expect X1, X2 are the same identical because they are random number generator inside the function. So you see the same shape, but the number should not be the same. Any questions for this? Very quickly go through function definition. So there are a lot of functions which not in your working space, but if you see, okay, which function are in the NEs, so you can find, okay, this is a built-in function located in here. But if you see, okay, which my function, this which function will tell you there's a function called my function is located right here in your working folder. So you can find and to make a change. So now we are going to make a change for this my function. As I said, we want to generate, for instance, not only 5,000 cells, because now I'm not giving any chance to change the number of cells. But if I want to generate only 500 cells, I cannot do it because my current definition of the input does not allow me to change this to 1,000. This will give error message. So I will have to reopen my function definition and introduce a variable to allow this function to take this integer input in order to take that variable. So here I'm going to give a number n. So this expression tells the function you can take n as an input. So this n will replace okay. So now my function require n as input. but I still have 3 genes will be generated and put them together. So now if I call my function again, now this time, this x, this time the function read, okay, you gave a number, so this number determine how many cells I will generate. Okay, any questions for this? Okay, so your function, you have to consider how can you take the input variable and make changes and achieve the computation and generate the output and give the output to there. So that's the making of a MATLAB function. Can I do this? Just one. Is that possible? This is fine, just one cell. And I can run that again. So this is just one cell each time compute. I can 2 cells. Okay. 3 cells. 300, they're all the same, okay? Because you have a variable there. We could do this before when we not giving any parameters. It generated 5,000 cells. But that 5,000, the number was hard-coded. But right now, if you don't give any input, n will not be assigned. Then this will give an error message because the function need n to be there. So this time, because you change your function, become a variable dependent, so you cannot ignore that input variable. Okay, so we finish this part now. I'm going to simulate a real single cell sim data. Okay, so this is a real function. What you had is just a toy. You simulate 3 genes, then the number of cells you define. But this one, can you tell me where this function is defined, where it's located? So which we need to have which this function is installed or located in this toolbox in this M file. So that's exactly same as what you have just done, my function, but this function is I provided. This function is to simulate single-cell RNA-seq data. So how to use it? So I know, because I made it, so I know simulate data. And what you can do is to start to type that in, and with a bracket, it start to show you how this function should be used. There are some tips here. So the first one, we need the number of genes you simulate. In our simple example, you have 3. You cannot change, but now you can change this number. So you may want to give a number here. A number of cells, we tried 5,000, 1,000, 50, 1, it doesn't matter, okay? So you need these 2 parameters. Their method type doesn't matter. So we need 2 numbers, these integers. So give it a try, and for example, I simulate 300 genes in 500 cells. So output. So output is x, this x is 300 by 500. This is output we just created. So in the function, I just do a loop in the first gene, second gene, repeat 300 times. And each time I pop up 500 random number, follow the negative binomial distribution. If you open this function, you can do edit and look at the source code here. You will find the negative binomial random number generator function in there, which is not very much different from my function you just created. Okay? Any questions? So I open this function. You see this definition, and this function has to be named the same as the file itself. And that's the input variables. I have an explanation. This common green will be the free text. You write comments. Any number, so by default will be 500, 1,000, and there are two different methods you can use. X is output simulation, and their reference. Okay, depends on if you use a simple method, and then you start to see this loop by how many genes you want to create, and you see this function where it's negative binomial. The parameter change every time, but this p-value 0.1 is kept constant. This shape changed, so average changed based on the gamma distribution. So this random number put in here, and this is fixed. So every time the average difference from one gene to another. That's all. So that's very simple. And so now we know the function you can simulate gene expression, and for any given number and genes, doesn't matter. So in this interface function here, if you click this import data, and you can see the simulate data, when you select this option, the simulation function I just show you will be called. So that's where this linked to that function will generate the matrix expression matrix, and will be given the name of genes, just like G1, G2, G3. There's no real gene name, but this is where you can call that function through the interface. So you can try, click this and OK, and it should ask you how many genes, how many cells you want to simulate. So by default, we have a 3,000, 5,000, and then the simulator is running and to generate the data. And then the data is put in here. That's 5,000 cells. If you look at the gene, you click the first buttons here, then you have G1, G2, all those genes. Okay. Do you want to take a break? Or you don't have to? You want to take a break? Okay. Okay, now we are going to simulate a matrix, gene expression matrix. Then we will see how can we compute in MATLAB to do this library size normalization for any given expression matrix. Then we will go through this descriptive statistics for gene expression mean, gene expression variance in the across the cell, standard deviation, and the coefficient of variance. Now let's use the function to generate an expression So this is a simulation, and generate an X matrix with 3,000 genes and 5,000 cells. So this is the matrix. Now, so what is the library size? Library size is the total number of UMI counts per cell, each cell, and that gave you the idea how deep it is sequenced. So you have to do is to take a column sum. So everything in each column sum together to give you a value. Let's call it lab, so it's library size. The function you want to use is okay. Lab Z, I gave a lab size. So you can check, make sure it's correct. If you look at this, if you double click this, you should see, okay, there are 5,000 numbers, each corresponding to each column, because we have 5,000 columns. Those numbers are library size for each cell. They are very similar to each other because our simulation does not create strong library size variation. Our simulator is too simple, very naive. So you don't see much. But in real data, you may see, okay, there are 5,000, there are 20,000, huge variation. In that case, you need library size normalization more than this case. Okay, but the code, the way to compute is the same. So now we have to divide each column. So the first column in the X, all the numbers will divide by the first element in the library size. So this first number is the library size for the first column. Second column, you divide by the second number in this library size. In MATLAB, it's very easy to do. Not in every language, but this is nothing for MATLAB. So this operation will do the process I just described. there are 5,000 numbers here, each column, and then every column in the X will divide by its corresponding library size element. Now, if you look at X and this normalized version of this is very small because you all divide by this total. So if you do some xn, so this normalized library size should be 01 because you normalize by itself, by the total number. So this should be 1 all the times, okay, in your answer. So that's what I did. Compute the normalized matrix, the library size, the 01 by itself. So this number is just too small, so people just that number is too small, so we just arbitrarily select a factor, 10,000, then times that, so reconstruct the matrix. Okay, so this is normalized version. This is a normalized version of that expression matrix by library size. Okay, I don't know if you can see my number 12 gene in first cell have a high expression, is 22. Well, my first gene is 0, so my number 22 gene seems to have a high expression. If you look at the normalized version, my 12 gene have 8. My first gene in the first cell is still 0. So everything is proportional, unchanged. There's only through normalization, I just made every column have a same summation, same total. That's all it changed. So so the gene, the number may change, but the overall pattern should be kept. So the number 12 genes still highly variable, highly expressed. Number 4 genes, and in my cell number 4 is 66, really high number. If you look at this normalized version, number 4 number for gene, the number for cell is 24, which is much larger than others. So everything is still proportionally enlarged or decreased proportionally. So everything keep the same. The only change is normalized by library size. Okay. So this is a library size normalization. We also will do a log transformation for the normalized version. So this is a log norm. So we finished the most commonly used library size normalization following the log 1 p transformation. So this combination, so library size plus log transformation, is mostly commonly used pre-processing steps to give you this norm transformed matrix for the downstream, for the clustering, for dimensionality reduction, for correlation analysis. They all acceptable. So this is the way to do. So let's just repeat what we did. So we have this unnormalized row matrix. Of course, QC had already been done. Okay, all the very extreme empty genes have been removed. Empty cells or poor quality cells will be removed. So this is a kind of QC'd, basic QC'd expression matrix. But after that, we did this library size computation. So we have a lib size. Then we did xn. Then we increased by a factor 10,000. And then, so this part is this library size normalization. And right after, we can have this log 1p transformation. Then we are done. So from the beginning, from the QC'd X matrix, we use 2 lines of code, finish the most commonly used normalization and transformation. Okay? There are other ways to do this procedure, but this is sufficient most of the time, will be enough. Okay? This factor you can change there, different, because this is only change the scaling, but not the relationship between the cells, between the genes. So this really doesn't matter. If you put 50,000, still have a same results in end. Okay, any questions for this part? Okay, now we will be focused on, we can use normalized version. We can play now, put this aside, x, but we're now working on xn, normalized version of xn. Now you compute the average expression of gene. So you have xn. So now we compute it in terms of gene-wise. Okay. Gene-wise, because cell-wise average should not change because we already normalized the total summation of the cell should be the same. So the mean for the cell for this normalized version means nothing because all the cells have the same mean. But for the gene-wise, it's meaningful. So now we focus on this normalized version, compute the average genes expression average. So the function we will use just mean. So this function, so we are going to compute mean for our nxn the 2, okay? So if you don't put, it's too small, right? So let me make the font bigger so you can see. If you don't put 2 here to tell the function that we are going to by row, compute the mean by row, by default it will compute the mean by column. That will be the total library size, average library size for the cell, because we already normalized that the mean should be the same. So we have to give the indicator to say, okay, we will compute average by rows, not by columns. So any questions for this part? So we have U, this mu, and this average expression for genes. How many genes we have simulated? 3,000 genes. So this mu is supposed to have 3,000 numbers in there. So we can see first gene have a 0.5 expression average across those cells. Gene number 8 is 1.2, so higher expression. Gene number 9 have a 2.0, so higher. So this is compute variation, so V, okay? The function is called a VAR. Variation and also to indicate for the genes. let's see. Let's call this V2. Yep, I changed the variable name. I want to use this as a standard deviation. So V is standard deviation. So similar, okay, very similar calling of function. This particular empty parameters. So if you look at the documentation for the function variation, so there's a second variable W. So this is by default is a 0 or 1. So if you give nothing, it will be, this second variable will be 0. It indicates that when you do the normalization, you will have n minus 1 in your, when you compute the variable. Okay, now we have standard deviation, which is V here, standard deviation. We have a mean, U here. So we can compute the CV. So CV is standard deviation V divided by mu, okay? We just follow the definition. So standard deviation divided by the average. You compute the coefficient of variance. So now we finish this part, okay? Gene-wise average, mu, variance, standard deviation. Standard deviation is the square root of variance. CV is a standard deviation divided by mean. We have those. So those are the definite, the gene-wise statistics. So each point is a gene. When you compute this point gene, there's 3,000 numbers for the variation, standard deviation, 3,000 standard deviation, coefficient of variance, variation for the genes. Now we are going to plot a scatter plot, something like this, for gene-wise. So we are going to put mean here, which is a mu, the value. We're going to put a CV here. Okay? In this scatter plot, each point will be a gene. So if we have a gene, have this particular mean corresponding to CV here, we will put that gene here. And another gene, if we have a higher mean, and it will be that end, lower CV will be here. If a gene have a lower average but a high CV, we'll put it here. So we will generate a scatter plot to look at the relationship between those genes, relationship between their average expression and their CV. So to show this two variables relationship. Let's make a figure. So we're ready to have this canvas to allow the figure to be in there. So the scatter, we will put a mu and CV. So this will put the values in average expression and the CV. So you have a 3,000 each on here, okay? there are many dots, 3,000 dots, and they are put onto each other. So you look like the curve, but if you look at it carefully, they're circle dots. And they follow this beautiful curve. There's one gene, for example, this one, have very small average, but a really high CV, and then there another one. But for the gene expression level, on average, it's high, so this particular genes in this range, on average, they have a very low CV. So these two variables to characterize genes' average expression and their dispersion are linked somehow, okay? So their average and the CV is follow this particular curve. You can click this to get values for those selected points. You can see this relationship. You can do X label with a string, so it's a mean, and you put a Y label as a CV. Then come back to this, you can see you add labels for them. So now this is the average, this is CV, and the points are the genes. Yeah, so use this version. So this is a log-log plot. So use this version to indicate. I will use a circle to represent every gene. If you're not, then it have a link between genes, the lines. So in this log-log transformation plot, the average and the CV, the values have been transformed into the log scale. And then you see almost a straight line here. Okay, that's a relationship between these two variables, which is this part here, okay? These two descriptive statistics for the genes, cross gene populations. These two variables are linked on the log scale as a straight line. So if you have a high expression, you should have a low CV. So highly expressed gene, the variability you measured should be lower, because you don't have so many zeros in there. If you can imagine it that way, then the variability measured by the CV should be lower. So this makes sense, but the straight line still indicate there's something that in our simulation, because we follow this Poisson negative binomial, so we generate a data set which almost perfect follow the theoretical distribution for those relationship between CV and the mean, which given by this relationship, because our simulated number essentially follow the Poisson distribution, okay? So there's a relationship between mean and the variation. Because of the random number generator we use, they're somehow linked. So at the end, we recover this theoretical relationship which is straight line between log scale CV and mu. Okay, we need to move to a calculation of the variance and the covariance. Okay. We are going to use examples. So now we can clear this. We are going to use example here to compute. Let's compute the variance first. So you need to type this x variable numbers in. So you create a vector and then called x and put those numbers in. So we are going to compute the variance for variable x. So my x is okay. Any questions for this part? No questions, so I just type that. So now we are going to compute the variance for this. The variance indicate how dispersed of those data points in that variable away from the mean, right? So we need to compute the center of this. So mean x. Okay. So now we need to have x minus that mean, which is the distance from your center. So let's see one step by each. So mean is 14, which is this one here. And your x itself, every element minus this mean, create this column. Then we take a square for them. Okay, so the expression I'm writing here is this column. And the 3 become 9, negative 7 become positive 49. So element based, okay? Every element in your variable minus the average of this variable, create this distance, then you squared and get basically this area for those. Okay, so then take them together, sum, to get 166. So 166 divided by 6 minus 1. Because you have 6 samples in your data, so that's the variance compute. So let's redo it. So my calculation for this. So my calculation, step by step, follow the PowerPoint slide on that page is 32.2. I can also use this function, which built-in function to compute the variance. And you get the same number, okay? Now we move to covariance. Covariance is between two variables. You have x, you know how to compute the variance for x itself. You know how to compute variance for y by itself. Just use the same procedure up here, but the relationship between these two is called covariance. You have to do in this way. So let's type this in. 5, 8. My y is okay. I have my x and y. I want to compute the covariance between them. The first, I need to use x minus its own mean to get the centralized, okay? X minus mean x, cxi, xc, okay? You see the same number of this column with that column. We do the same thing for y. So y centralized y minus its own mean, this column. So we have cx, cy of these two columns, okay? YC. Now we need a product of these two. XC times YC. I got an error because this becomes a matrix multiplication, and then my inner product doesn't match. So x is 7, 8 by how many? 9 by 1, 9 by 1. So it's not doable. So this is element based. So we need to do dot, make product. This is the last column, the product by element. So that's what we did. Okay. So the results of this, the whole column will be add them together to get this total. That's the final result we get. So I can rewrite everything from the beginning. So this x minus mean x multiplied by y minus mean y. This is a single line expression for the whole table in the slide. Any questions for this? So every variable centralized by minus its mean, and then you make product between elements in these two variables and sum them together. So this number is divided by the sample size, because this number will be getting larger and larger if you have a large number of samples. So you need to divide this to a total 8, so you get this covariance value. Okay, this whole expression, you can use covariance function x and y to get something like this. What you found is this covariance function gave you the covariance matrix, which in the diagonal position, upper right and bottom left, this is a covariance between these two variables, which is same as we compute. And in this diagonal position, this off diagonal is pairwise between x, y, the covariance will. This is a variance of x, and this variance of y. Okay. So if you compute variance of x, it should be this, and the variance of y should be that. So this particular function will compute each variable for each element, each variable itself, and put their variance in the diagonal position where the off diagonal gave you the value of covariance between variables. Yeah, you can give the try for this like 3 variables, and in the A you have 1, 2, 3 columns. This is like 3 x, y, z. So we only have a x, y as an example here. But if you have a x, y, z, you should have a 3 by 3 covariance matrix, because you have 3 variables. There are pairwise relationship between them. So you can try this method, and okay. So if I do dot x, y, I try with what I can get. Covariance matrix. Yeah, that's a different. Okay, so let's see what left in our outline here. Okay, in the last 5 minutes, I will skip this and very quickly show you how to do the differential expression analysis in the interface. There's no, it's all just by click. Okay, so we need 2 data sets in order to do the comparison. So we will import the data from a geodatabase. And we need a number. I will type this in, and then 49. 8, 5, 4, 9, comma, 55. So 2 samples. you need 2 samples in order to do a comparison. So I put this accession number. I know they all exist. And then the 2 samples will be download and merge. I can't remember exactly the human. Let's just assume they're human. Okay, so let's take a look of the batch ID. I want to see the difference. They're blue and the red cell, they're from a different sample. One is from this 49, another 50. Let's see there. Looks like they are mouse samples. So I will do my filter to get the QC. Should have already been done. I will remove some genes. Okay, make some clean for the genes. Mouse gene. So I have a smaller number of genes. Okay, in theory, we should do the annotation and to compare the same type of cells. But I just very quickly just show you the next step. We can use analysis to find a differentially expressed gene by doing this DE analysis. You have to select a working folder where you want to save your results. Then the comparison is between the batch ID. So you compare these 2 samples. You select both and then who as a reference. It really doesn't matter, and you can interpret the results later. So this is all you have to click to get the differential expression analysis. These 2 batch of cells will be compared. Their average expression for all the genes included here is we have 14,000, and then the results is here. Can do some of this next level of analysis, but at the end, you should get this kind of volcano plot. So to
to get the results. So this is a highly expressed upregulated, this is downregulated. Compare these two samples. Then you have a summary Excel table which gave you the upregulated, downregulated gene name. Okay, so that's all you need to do very quick DE analysis by compare two samples. Okay, so I haven't done this correctly because the cells are highly heterogeneous. So we supposed to find only one cell type to compare, but now it's just globally compare the average between two samples. Okay, so this is all for this week. I can take a quick question before we finish today. Otherwise, see you next Tuesday, and there will be an important lecture for PCA analysis.